All right. The question is, how do substances get into the cell or out of the cell? And so we're going to review right now diffusion, osmosis, active transport, passive transport, and so on. We're going to start with diffusion. Now, diffusion, I wrote, is the spontaneous movement of a substance. Chemical substances are also known as solutes. So it's the spontaneous movement of a substance down its concentration gradient. Chemicals spontaneously flow from an area of higher concentration to an area of lower concentration. Let's look at a picture to help us. Look on page B25. I wrote that, B25. And on the bottom of B25, all right, so the little picture is labeled diffusion. So here it shows a whole bunch of, and I wrote oxygen. All right, O2. Oh, these are O2 molecules. And there's a whole bunch on the outside of the cell, and there are fewer oxygen molecules on the inside. So chemicals tend to flow or diffuse from an area of higher to an area of lower concentration. Now, no matter how much oxygen flows into the cell uses the oxygen up. It uses it up, so it's always flowing in. How does a cell use up oxygen? What's it doing with oxygen? Very good. It's attaching the hydrogen atoms from the foods that you ate that you're breaking down to make ATP, such as sugars. It's attaching those hydrogens onto oxygen to form water. That's how the cells dispose of these hydrogens as they break apart sugars and fats to generate ATP. So they're always using up that oxygen by turning it into water. So it's always flowing in. Oxygen is a very small molecule. It's just 2 atoms big. It can easily get across the fatty cell membrane, just 2 atoms big. An example of a chemical that's always diffusing out of the cell is carbon dioxide, CO2. Carbon dioxide here, it shows a whole bunch inside the cell. The arrow indicates that it's diffusing or flowing out. Simply flowing from an area of higher concentration to an area of lower concentration. Where's all this carbon dioxide coming from? Exactly, cellular respiration. We break apart sugars and other foods into carbon dioxide. And the hydrogens from those foods were attached to oxygen to form water. This is why you need oxygen. This is where carbon dioxide comes from. It's all tied in with cellular respiration. All right, now, just going back to where we were on page B23. On B23, we're talking about diffusion. And what makes these atoms and molecules move? We know that they're going to move from an area of higher to lower concentration, but why are they moving? And the answer is kinetic energy. All atoms and molecules are in motion. You'd say what? The tabletop that's right, your tabletop that's right in front of you, it's made out of atoms and molecules. Are they moving? They are moving. They're vibrating. Everything, all atoms and molecules are in motion. And so what happens, because they're all vibrating and moving, where they're concentrated, they bump into each other. And as they bump into each other, that causes them to spread out. Now, what would be a way of slowing down the rate at which they're moving? Lower the temperature. So the rate at which these atoms and molecules are vibrating and moving at, and thus diffusing at, is affected directly by the temperature. So we wrote that on page B27. On page B27 at the top, B27, so increasing the temperature increases the rate of diffusion. I think you'll learn in a chemistry class that for every increase in 10 degrees centigrade, you get a doubling of the rate of chemical reactions. Does that sound right? Right? That's the rule of thumb. So you increase the temperature, increases kinetic energy, and makes things move faster, bump into each other faster, diffuse at a faster rate. The converse of that is lowering the temperature slows down the rate of diffusion. How low do you need to lower the temperature to stop all atomic and molecular motion?
0.
Okay, well, 0 on what scale?
Celsius.
Okay, so 0 degrees Kelvin, minus 273 degrees centigrade. What's that commonly referred to? Minus 273 degrees centigrade? Absolute 0. All right? And so the lower the temperature all the way down to minus 273 degrees centigrade slows down atomic and molecular motion. That's called absolute 0. All right, now there's some other factors that we want to mention also that can affect the rate of diffusion. Let's go back to page B23. So that's what we wrote here. Factors that affect the rate of diffusion. So we know, we've already mentioned temperature does that. Here's another factor. So I drew a picture of two cells. Pretty magnificent pictures. All right, so here's two cells. And on the outside of this top cell, we indicated there's a whole bunch of some molecules there. And none of those molecules inside the cell. In the lower picture of a cell, we've similarly got a high concentration of some molecules on the outside, but there's already some of them on the inside. Which one of these, the top picture or the bottom, has a bigger difference in concentration between the outside and inside? The top one. Here, there's a bigger difference in concentration. Where will there be, the top one or the bottom, will there be a faster rate of diffusion? The top. The bigger the difference in concentration, the faster the rate of diffusion. So when there's a big difference in concentration between the outside and inside of the cell, the rate of diffusion is faster. Where there's a smaller difference in concentration, the rate of diffusion is slower. So that's another factor that affects the rate of diffusion. Another factor, very easy to understand, is the size of the chemical that's moving. Now, here I've drawn 3 different size molecules. These are not meant to be really realistically depicted relative to a cell. Here's a cell. So I drew a real tiny chemical, a medium-sized chemical molecule, and a large chemical. And what's an example of a really small chemical or molecule? O2. It's 2 atoms big. What's an example of a molecule that's medium-sized? Glucose. C6H12O6 is 24 atoms big. What's an example of a really big molecule? Protein, which is thousands of atoms big. Hundreds, more than that. All right. Now, these are drawn relative to each other, but not proportional to the size of the cell. So which one of these do you think diffuses across cell membrane fastest? Yeah, the smaller. That's pretty logical. So the smaller the molecule, the faster the rate of diffusion. The larger the molecule, the slower the rate of diffusion. Obviously, you reach a certain size where it just can't get across the cell membrane at all. In cell, and that's the case of protein. Can't get across at all. Sugars can get across, but not very well. That's why they have to usually be helped out by a transport pump. All right, on page B27, we've already mentioned on the top of B27 that temperature, the amount of heat or kinetic energy, the temperature affects the rate of diffusion. And the fourth factor is whether the chemical that's going across the cell membrane is water-soluble or lipid-soluble. Let's imagine we had 2 molecules that were the same size. I don't know if my picture makes them look the same size, but that was my intent. So imagine we've got 2 molecules that are the same size. One of them is fat-soluble, lipid-soluble, the other is water-soluble. Now, before I go any further, you might ask, what does that mean, lipid-soluble or water-soluble? Which chemicals are fat, lipid-soluble, which ones are water-soluble? Any chemical that is a fat, that is a lipid, is lipid-soluble. there's an old expression in chemistry, like dissolves in like. Have you ever heard that? All right, so a fat dissolves in a fat. If you have peanut oil, will it mix with corn oil? Absolutely. Have you ever heard of blended oils? Don't they have a number of different kinds of oils mixed together? So you can mix any vegetable oil with any other vegetable oil. Any fat with another fat. All right? So anything that's a lipid is lipid soluble. Anything that's not a fat is generally water soluble. You'd say, yeah, like what's an example of something that's not a fat? How about sugar? All right? Will sugar dissolve, let's ask this, will sugar dissolve in vegetable oil? No. No? Try pouring it in, it won't dissolve. All right? Will sugar dissolve in water? Absolutely. All right? So sugar is water soluble, it's not a fat. An amino acid is not a fat, it's water soluble. So we've already talked about vitamins. We talked about water-soluble vitamins and fat-soluble vitamins. So anything that's not a fat is water-soluble. So which one of these do you think gets through this fatty cell membrane faster? A fat-soluble molecule or a water-soluble? All right, well, I tried to cue you in. Which kind of molecule gets through a fatty membrane easier? Something that's fat-soluble. Because fat dissolves in fat. So anything that's a fat tends to get across that fatty membrane. Remember, the whole middle part of a cell membrane is a double layer of fat. So two molecules that are the same size, the fat gets through the cell membrane faster. A couple of quick examples of that. Now, gasoline comes from oil. You know why they call oil oil? Because it's oily and greasy and fatty. All right, gasoline's made from it. So if you, not uncommonly, sometimes when you're filling up your car with gas, you get some gas on your hand from the nozzle as you're handling it, right? So you could, smell the gasoline on your hands. So you figure, I'll go home and I'll wash my hands off with soap and water. That'll get rid of the smell, does it? Usually not. You know why? Because gasoline is so fat soluble, it penetrates cell membranes very quickly. So it's deep in your skin. That's why it's not going to be removed by soap and water. Because something that's a fat gets across cell membranes faster, anytime you take a medicine and you want it to penetrate into your skin, it's always going to be in the form of a cream or an ointment. Creams and ointments are fat-soluble bases. Right? Because that helps it get through the cell membranes. That helps it penetrate your skin rapidly in the form of an ointment. So everybody has put like Neosporin or Isotonic on your skin, right? Isn't that in the form of an ointment? It's greasy, right? That helps it penetrate, being in a fat-soluble form as opposed to water-soluble. That's why the reason why we do what? Well, we get fat mostly because of eating too much Chunky Monkey ice cream and not exercising. Huh? I'm sorry, not me. Yeah, I'm sorry.
Nothing. I don't know, because I thought maybe our cells get bigger.
Well, your fat cells can accumulate more and more triglycerides. That makes them bigger. But we're not usually eating by smearing fat on our skin. I don't know. I usually eat the Chunky Monkey ice cream. I don't usually smear it on my skin. Okay. So we've been talking about 4 factors that affect the rate of diffusion. Now, next thing we want to talk about is osmosis. Osmosis. And osmosis, ostensibly, is simply the diffusion of water through the cell membrane. We could just say diffusion of water. We don't have to say the word osmosis. Why did they give it a special name? I think the reason why the diffusion of water, when water diffuses into a cell or out of a cell, the reason why they give it a special name is it's the only chemical, biologically, that is a liquid flowing across cell membranes. All the other chemicals are solutes or gases. They are solids or gases. Water is the only liquid that's usually biologically moving across the cell membrane. But it works just like any other solid or gas. It flows from an area of higher to lower concentration for that molecule, in this case water. Now, to help us understand this, let's look at the bottom of B26, the previous page. And we'll walk you through this. All right, this is a red blood cell. The fact that it's a red blood cell, it doesn't have to be a red blood cell, it could be a liver cell. It could be a pancreas cell. So we're using this red blood cell as an example of any cell in our body. It represents any cell. Now, we have already learned that most of what's inside of a cell is water. It's 80% water on the inside. We mentioned that before? Isn't the cytoplasm? No, our body is 60% water, but a cell is about 80% water. You have it in your notes. I already know what page. Look at page B1.
I know, but H2O plus 14.
It's 80% water by weight, and then the next major chemical is protein. All right, it's page B1. You can check it. What's another name for cytoplasmic fluid? Page B1, ICF. What's ICF stand for? Intracellular fluid. All right, it's mostly water. And then after water, there's proteins. But it's not just proteins inside the cytoplasm. Protein is the major chemical by weight, but there's sugars, and there's vitamins and minerals, and all kinds of other solutes, all kinds of other substances in that fluid. So I kind of colored this bluish to represent the water, and these dots represent the solutes, the chemicals that are mixed in with the water. Proteins, sugars, minerals, vitamins. That's what those dots represent. Now, if the fluid, if the extracellular fluid, the fluid on the outside of the cell, is also 80% water and 20% other stuff, then the concentration of water on the outside of the cell is the same as the concentration of water on the inside of the cell. So if the concentration of water in this fluid on the outside of the cell is the same as the concentration of water on the inside, we call this fluid on the outside isotonic. What does iso mean? Same or equal. So this fluid on the outside of the cell is the same tonicity, the same strength, the same proportion of water and solutes. You'd say, what are you saying, water and what? I'm saying water and solutes. Solutes are what's dissolved in the water. It just means chemical substances. So if the proportion of water and solutes on the outside of the cell is the same as on the inside of the cell, we say that that fluid on the outside of the cell is isotonic to the inside. Now, if the concentration of water is 80% on the outside and it's 80% on the inside, is the water going to rush into the cell? No. Is the water going to rush out of the cell? Why not? Because the concentration is the same. So there's no diffusion. There's no net diffusion occurring. What's the term we use for the diffusion of water? Osmosis. There's no osmosis. This is a happy cell. Look at that. Smiling. Because the cell is not swelling up with water and it's not collapsing as the water goes out. Now, alternatively, let's consider another scenario. What if we have a cell, starts out 80% water on the inside, 20% other stuff, but this time this cell is surrounded by distilled water. Pure water, 100% water. 100% water. Now, what's water going to do? It's 100% water on the outside, 80% water on the inside. Is there a concentration gradient for water? Yes, so the water starts to flow into the cell, down its concentration gradient, from an area of higher to lower concentration. As the water starts to flow into this cell, the cell starts to swell up, bigger and bigger and bigger. Now, it can only swell so much before it ruptures. What do we call when? What's the word for breaking apart? Lysis. When a red blood cell breaks apart, we commonly call it hemolysis or hemolysis. Hemo just means blood. But the bottom line is, whether it's a red blood cell breaking apart or any cell, you place a cell in a higher concentration of water than what's in the cytoplasm. The water flows in, there's osmosis, and the cell swells up and bursts. A fluid that has a lower amount of solute than normal is said to be a hypotonic solution.
You'd say that again?
We said an isotonic solution is 80% water, 20% other stuff, solutes. 80% water, 20% solutes. A hypotonic solution is higher in water and lower in solutes than the cytoplasm. Lower in solutes. It's low in protein, it's low in sugar, it's low in everything, it's just a bunch of water. Hypotonic, low strength. Low strength. So placing a human cell in a hypotonic solution, hypotonic fluid, will cause the cells to swell up and burst. That's not going to be a happy cell, I don't think. That's going to be a dead cell. Now, let's consider the third possibility. Third possibility. We've got a cell. It starts out 80% water on the inside, 20% other stuff. But this time, we're going to place this cell in a hypertonic solution. High solute concentration. It's concentrated in salt. That's what it says. It could have been concentrated in sugar. It could have been concentrated in protein. It could have been concentrated in a combination of sugar and salt and protein. The bottom line is there's not much water. I made up an example. Imagine this solution is only 20% water, 80% solutes. Sugars or salts or proteins or a combination of those, but it's only 20% water. Now, if this cell is 80% water on the inside, and now it's surrounded by a solution, a fluid, hypertonic, that's only 20% water, really high in salt, is there a concentration gradient for water? 80% water on the inside of the cell, 20% water on the outside. What's water going to do? Diffuse out. All chemicals flow from an area of higher to lower concentration. And as the water flows out, what do we call the movement of water when it diffuses across the cell membrane? Osmosis. There's osmosis. And as the water flows out of the cell, this whole cell is shriveling up. The term for that is crenation. It's undergoing crenation. It's shriveling up. That's not going to be a happy cell either. This cell burst, and this cell is shriveling up and collapsing. So, in general, and we're going to learn more about this yet in this course, anytime a solution is injected into the body, it must always be an isotonic solution. Always. Because if you're giving any fluid, any solution, intravenously into the body, a shot, it better be isotonic, or you're going to cause the water, if you're giving a hypotonic solution, the water is going to flow into the cells and burst them. Or if you're giving a hypertonic solution, the water is going to come out of the cells of your body and they're going to collapse. We're going to learn more about that. It's a very important subject. So all the most commonly used solutions in a hospital setting or a clinical setting are isotonic. So maybe you've heard of some things. Anybody heard the expression NS? Anybody know what NS is? Have you heard it called normal saline? Normal saline is an isotonic salt solution. We'll learn more about that. Yep. No. Interestingly, this only applies to injection. All right? Because that's going right into your body. You can drink you can drink plain water. You can drink salty water, soup. All right? It doesn't matter in terms of what we ingest, because we can absorb varying amounts of water and salts and so on. But if it's injected, it's not a matter of differential absorption of water or salts. It's all, whatever you've given, that's what you're giving, surrounding the cells with. So it only applies to what's introduced directly into the body by injection or IV drip or something. Those have to be isotonic. All right. So let's summarize what we've said about osmosis. On B27. So osmosis is simply the diffusion of water through the cell membrane. The water does tend to flow through protein channels. Some of it can very slightly even get through the fatty membrane, because water is a small molecule. There's a general rule that water follows salt.
You'd say, what does that mean?
All right, well, I'll explain this expression, water follows salt. Let's go back to our picture on the previous page. Technically, water is simply flowing from an area of higher concentration of water to an area of lower concentration of water. Here, water is flowing from 100% water flowing into the cell where it's 80% water. But, in a sense, you can think of the water being drawn into the cell where there's a higher amount of salt or solute. Technically, it's just water is flowing from an area of higher concentration of water to lower. Conversely, here you have a cell that's 80% water, and surrounding it, it's only 20% water. So there's actually a higher concentration of water on the inside than there is on the outside. All right? But in a sense, even though it's not technically correct, you can think of the water as being, in a sense, drawn towards where there's more salt. Here's a vivid way to understand what I'm trying to make. Has anybody ever poured salt on a snail? I got to tell you, I've been asking this question for 35 years, and I'm always astounded by how many people have done that. I always figure one semester I'm going to ask it and nobody's done it, but it's never happened. There's always people who've poured salt on a snail. Frankly, when I want to kill snails, I pick the snail up, I throw it in the street, and watch cars run over it. Well, isn't that instantaneous death? Here's what you're doing when you're pouring salt on a snail. When you pour salt on the snail, have you ever done that and you notice it's starting to bubble? Ever see that's like fizzling and bubbling and so on? You are surrounding the cells of the snail with all this salt, and there's no water. So in a sense, the salt is drawing the water right out of the cells of the snail's body, and all that water that you see bubbling out, it's literally all its cells are collapsing right in front of you. All right? So, I would just throw them in the street and watch cars run over them. But sometimes I'll roll them into the street, and they're trying to get out of the way, and they have no chance. The cars are. You like snails? I'll bring you some of mine. All right, so that's really what's happening, is that these, in a sense, the salt is drawing the water right out of the cells of the body. All right, so that's crenation. So the water is drawn towards the salt. Wherever there's more salt, that's where the water is drawn towards. Isotonic means that the concentration of water and solutes is the same on the outside of the cell as the inside. Hypotonic means that the concentration of water is higher and the concentration of solutes is lower. Lower solute concentration than is found inside the cell. So water flows out of the cell, water flows into the cell from the hypotonic fluid, high in water, low in solute, causing swelling and rupturing, lysis of the cell. And a hypertonic fluid, just like pouring salt on a snail, is that the water comes out of the cells towards that higher salt or solute concentration, causing shriveling up, crenation, collapsing of the cells. These are all very important clinical subjects we'll be learning more about. Let's take a look at page B29. On B29, so we have been reviewing diffusion and 4 factors that affect the rate of diffusion. We've reviewed osmosis and the terms isotonic, hypotonic, and hypertonic fluids. So now let's remind you about transporter proteins. Transporter or carrier proteins transport chemicals. They help transport chemicals across the cell membrane. So we wrote that there are proteins embedded in the membrane that transport specific chemicals. There is specificity through the membrane using energy supplied by ATP. Well, it uses ATP if it's active transport. If it doesn't require energy, doesn't require ATP, it's called passive transport or facilitated diffusion. Let's focus on active transport for a moment. Active transport processes in the cells of our body account for 40% of all the energy, all the ATP used in our body. At this moment, as you're sitting in your chair, 40% of all the ATP being produced by your cells is being used for active transport. So that's a significant amount. Now, what are you actively transporting? All right, these active transport proteins are also known as pumps. So they call another name for an active transport protein that's using energy for this protein to transport a chemical across a cell membrane are commonly known as pumps. So there are sodium ATP pumps that actively use ATP to pump sodium ions, potassium ATP pumps, iodine ATP pumps, calcium ATP pumps, hydrogen ion ATP pumps. In almost all these cases, the ion is being transported against its concentration gradient. That's why energy is needed. Energy in the form of ATP is being expended to force a chemical to go in the direction opposite to what it wants to go in. Let's look at a picture to help us understand this. On the previous page, on page B28, this is kind of a diagram. This represents a cell. Here's a cell. And first of all, we are reminded about ion channels. We talked about that last time. Ion channels are simply tube-shaped proteins that can open or close. And most of these ion channels, most of the time, are they open or closed? Most of the time.
Closed.
They're usually closed, but they can be open. And these, when they do open, they would allow ions to simply flow to diffuse down their concentration gradients. And when sodium ions open, sodiums usually just diffuse or flow into the cells. When potassium ion channels open, potassiums tend to flow out of the cell. When chloride ion channels open, chloride flows in. When calcium ion channels open, calcium flows in. We're going to be learning more about that. Those are just ion channels. Now, we've also we've mentioned facilitated diffusion. Facilitated diffusion is also known as passive transport. You might say, when did we talk about that? We didn't do that. All right, let me just show you. If you go back to page B26, did we do B26? Anybody have any notes there or anything? All right, we did it then. So we were mentioning these transporter or carrier proteins. If they use ATP to force a chemical to go in the opposite direction that it wants to go, that's called active transport. Now we also have learned the term that it's called pumps, active transporter pumps. If there's a transporter protein that just helps a chemical go across the cell membrane in the direction that it already wanted to diffuse in, it doesn't require any energy in the form of ATP, and that's called passive transport or facilitated diffusion. This is exactly what happens in the transport of sugar. Last class meeting, we looked at this picture. I think there was one or two people who didn't have this. Page B24A. Is somebody who's missing this? B24A. Anybody else? All right, so on page B24A, we talked about this last time. I'll just remind you. This is basically showing how insulin works. This comes from Wikipedia. I was just trying to get a diagram. So here's insulin attaching and activating an insulin receptor site. That in turn activates a slinky. That's the G protein. The slinky or G protein is attached to a glucose transporter protein. This transporter protein, or carrier protein, starts moving sugars, glucose molecules, into the cell. Now the glucose is flowing down its concentration gradient. It's just that glucose is a little bit on the large side to get easily across the cell membrane, so this transporter protein helps move it across. This is an example of passive transport or facilitated diffusion. A transporter protein is helping to move a chemical across, but no ATP is required. Once the glucose is transported into the cell, the glucose can be broken apart into pyruvate sugar, right, by glycolysis and used to make ATP, cellular respiration. Or it could be stored, the glucose molecules could be snapped together to form a polysaccharide called glycogen. All right? So we said that the importance of insulin is without insulin, the sugar that's in the bloodstream can't get into the cells of the body. So remember we said in diabetes, when you eat foods containing sugars, you absorb that into your bloodstream, you get high levels of sugar in the blood, but without insulin, it stays in the bloodstream, it accumulates in the bloodstream, you develop hyperglycemia in the bloodstream, and in the meantime, the sugars can't go into the cells. It can't be stored as glycogen, it can't even be used for energy. So in diabetes, the person's cells have to resort to using more fats for energy and proteins for energy because they can't use sugars. But this was an example of passive transport or facilitated diffusion. Is everybody okay on that? Covered that last time. So back on page B28. So that, glucose transport is an example of passive transport or facilitated diffusion on the top of B28. Now, there's also active transport. This is where ATP energy is used to force chemicals into or out of the cell against their concentration gradients. And this accounts for about 40% of all the energy used, generated by the cells of our body. ATP is used to force hydrogen ions out of the cell. ATP is used to force calcium ions out of the cell. ATP is used to force sodium ions out of the cell and force potassium ions into the cell. These are examples of active transport of ions. And these are also known as pumps. This would be called a calcium ion pump. This would be called a hydrogen ion pump. And this one here, where it not only forces sodium ions out, but potassium's in, is famously called a sodium-potassium ATP pump, because it's involved in actively transporting both sodium out and potassium in at the same time using energy. We'll have more to say about that as far as the ratio of sodium to potassium, but we'll get to that. Now, I also want to briefly mention something else. It's covered in your book. I'm not going to make a big deal out of it, but I do want to explain it. Sometimes the cells use energy, ATP energy, to force one chemical in, and as it forces a chemical in, another chemical just kind of follows. An example of that is when there's an active transport pump that that actually forces sodium ions in, and at the same time, let me give you another example. me give a simpler example of that. It's not shown here. It's not shown here. But let's imagine we had a pump where it pumps sodium ions in or out of the cell, and a chloride ion just follows. You'd say, what? Let me give you an analogy. Now, there are, not so much in Los Angeles, but in New York, there are department stores that have these kind of turnstile doors. I don't know, must be some in LA, right? Maybe in Beverly Hills or something? Anywhere they have these turnstile doors. Anybody know what I'm talking about?
Yeah.
Right, so you kind of push through. Now, imagine that I'm pushing my way through this turnstile door. I'm pushing in, and as I exert the effort, somebody comes in right after me, and I'm doing all the pushing. So I'm doing the pushing, and another person just kind of follows right after me. They're moving in the same direction into the store, or out of the store together. That is called symport. Sym means together. So they're being transported. The energy is being used to push one of them in or out, and another chemical kind of follows. So if the active transport pump was pushing sodium ions in or out of the cell, and a negative charge chloride just kind of follows. Right? Because it's attracted to the net positive charge sodium. So we would just say salt's going into the cell or out of the cell. Really, the pump is forcing sodium in, and the chloride just kind of following. Does that make sense? That's called symport. Together, they're being transported. Now, there's the opposite. What if I'm pushing the door at that department store, and I'm going into the store, and as that door is spiraling around, there's somebody else who wants to go out. I'm doing the turning, and they just kind of come right in, and they go out. Does everybody follow that? I'm doing the pushing, I'm going in, and they just kind of go right through the turnstile, and they go out of the store. I'm the one exerting the effort. They just kind of follow as the turnstile rotates. That's called antiport. Anti, against. So one of them, if you use energy in the form of ATP to force one chemical in, the other chemical just kind of follows out. So an example is forcing sodium ions in might result in calcium ions just flowing out, or something like that. That's called antiport. So symport is when they're going in the same direction, energy is being used to force one in and the other follows. Antiport is where energy is being used to force a chemical in or out, and the other chemical goes in the opposite direction. Let's imagine that we have a cell, and we increase the amount of chemical on the outside of the cell. As we increase the amount of chemical on the outside of the cell, we're trying to see what effect it has on the amount of chemical on the inside of the cell. Now, if it's diffusion, if it's simply diffusing into the cell down its concentration gradient, you tell me what you think will happen. As the concentration of the chemical increases on the outside of the cell, so that there's a bigger and bigger difference between the concentration outside and inside, you tell me what you expect to see happen to the rate of diffusion. As we increase the concentration more and more and more and more. It should diffuse faster, should be more and more diffusion. The higher the concentration on the outside, the bigger the difference there is in concentration between outside and inside, that should increase the rate of diffusion. All right, what does that look like? It looks like this graph. Here's what the graph is showing. On the x-axis, it's labeled extracellular solutes concentration.
You'd say, yeah, what does that mean?
That means the concentration of a chemical, a solute, on the outside of the cell. Extracellular solute concentration. I made up numbers. 0, 1, 2, 3, 4, 5, 6, 7. Just to indicate that the concentration is increasing. Now, what does it show on the y-axis? On the y-axis, it says flux into cell. And if you haven't heard the word flux, you're thinking, isn't that a dirty word? We shouldn't say that word in public. Flux. Flux means movement. So it's indicating the movement of a chemical into the cell. And just to give it some numbers, I wrote 0, 1, 2, 3, 4, 5, 6, 7. You'd say, I still don't get it. We're simply saying as we increase the amount of chemical on the outside, from 1 to 2
2 to 3 to 4 to 5 to 6
to 7, higher and higher concentration, how will that affect the rate, the movement, the flux of the chemical into the cell? If it's simple diffusion, as the concentration increases on the outside, the amount of flux, the flow into the cell increases. Now, the way we read graphs, and if you've had chemistry, you should have learned this, but whether you did or didn't, you're going to learn it in this class. The way we always describe a graph is we always begin as the. If you didn't know that, you're learning it now, because we're going to see a lot of graphs in this class. As the. You'd say as the what? We say as the, and then we look at what's on the x-axis. What's on the x-axis is called the independent variable. The independent variable. So, as the what? What's happening to the extracellular solute concentration? It's increasing. So, as the extracellular solute concentration increases, now we look at what happens on the y-axis. What is the y-axis showing? The flux into the cell, the flow of the chemical into the cell. This, what's on the y-axis, called the dependent variable. Does this ring a bell for anybody? It's like in math and in graphing and in chemistry. All right, so this is the dependent. So, look at this graph line. As the concentration, let's just look at this. As the concentration increases, if it's this much, we get this much movement into the cell. If the concentration on the outside of the cell increases to 4, we get more flux into the cell. As the concentration increases more, we get even more flow of the chemical into the cell. So, as the concentration of a chemical on the outside of the cell increases, the flow of the chemical into the cell increases. It is a straight line or linear relationship. Linear means a straight line. All right? It's directly proportional. And that's typical of diffusion. As the concentration of a chemical on the outside of the cell increases, the flow into the cell increases proportionally. Directly proportional. It's linear. That's what happens in diffusion. Now, what if we increase the amount of a chemical on the outside of the cell, but it didn't do that? It didn't increase the flow, at least linearly. You'd say, what do you mean? What if instead we saw this happen? As the concentration was increased on the outside of the cell, initially the flow increased, the flux increased into the cell, and then it maxed out. Can everybody see here it's reached a maximum level? Even though we're still increasing the amount of the chemical on the outside of the cell, we're not seeing an increased flow in. Then it's not diffusion. You'd say, what do you mean? It means it depends on a transporter protein. You'd say, I don't get it, I'm lost. I'm going to pretend, because I'm a great actor, I will be a transporter protein. And my job as a transporter protein is to help move a chemical on the outside of the cell in. All right? So let's imagine we start to increase the amount of chemical on the outside, and my job is to move it in. I'm helping it. Right? So the concentration is increasing. All right? So, oh, it's increasing, so I'll work faster. Oh, it's increasing more. I'll work even faster. But at a certain point, I'm working as fast as I can. I can't help it any faster than that. When it relies on a transporter protein or carrier protein to help the chemical across, it always shows a maximum. Because that carrier protein reaches a point where it's moving, it's transporting as fast as it possibly can. It's basically working as fast as it can to move that chemical across, so it exhibits a maximum limit. That's always characteristic when we see a maximum limit, that it involves a transporter or carrier protein rather than just simple diffusion. Everybody follow that?
So is this in the book?
100% it's in the book. Absolutely it's in the book. So you might want to read a book. But that's characteristic, and you should understand what we mean by the x-axis or independent variable, the y-axis or dependent variable, and how to read a graph. You always begin what's happening on the x-axis and how does that affect what's showing on the y-axis. Okay, now as we wrap up as far as how things get in or out of the cell, two last ways that things can get into or out of a cell. Phagocytosis, also known as endocytosis, and letter E, secretion, also known as exocytosis. In both cases, ATP energy is needed either to bring something into the cell or to take something out of the cell, because it involves a change in the shape of the cell membrane. Let's show you a picture on the previous page. On the bottom of the previous page, this is showing an example of phagocytosis or endocytosis. Here is a white blood cell that is in the process of engulfing or swallowing up some bacteria. It is changing the shape of the cell membrane. This requires energy in the form of ATP for the cell to change its shape. By the time we get to the third picture, 3 of the bacteria that used to be on the outside of the cell have been swallowed up within a food vacuole on the inside. This is called phagocytosis, phago meaning to eat. The cell has eaten. Or endocytosis. The cell is taken within something internally. This does require energy. The very opposite of phago or endocytosis is shown right here on the bottom right. This is called secretion or exocytosis. This is where a vacuole moves to the cell membrane, fuses with the cell membrane, and releases what was inside the cell to the outside of the cell. This is called secreting a chemical, or exocytosis. Exo as in the word external, to the outside. This also requires energy in the form of ATP to cause a change in the shape of the cell membrane. So these are just the opposite. Here stuff is going into the cell, here stuff is going out of the cell. In both cases, it does require the use of energy in the form of ATP to make that happen. So we have now described the major ways that chemicals can get in or out of a cell. Now, before we get to the break, let's really summarize what the hell we've been doing for the last week or so. And many of us are thinking, yeah, what have we been doing? All right, so we've got a cell here. This is the cell. And we know this cell, this human cell, this, the liver cell, pancreas cell, whatever cell in the body, needs various nutrients to stay alive. And we know that all living cells produce waste products that have to be eliminated. What are some of the nutrients that the cells of our body need? So I wrote O2, oxygen, and C6H12O6, sugars like glucose. Why do cells need oxygen and sugars? For the process of cellular respiration. We have reviewed cellular respiration. Cellular respiration is the process by which sugars and other foods are broken apart with oxygen to produce, in order to produce ATP, the gasoline that powers cells. But we know that as the cells break apart sugars with oxygen, they produce waste products such as CO2. That's a waste product that has to get out of the cells. What other nutrients do cells need? They need amino acids. Why do cells need amino acids? What do they do with them? They join amino acids together to make proteins. That's called protein synthesis. Cells manufacture all these different proteins. There's cells that make hemoglobin, and cells that make actin and myosin, and cells that make collagen, and cells that make keratin, and cells that make antibodies, and cells that make and secrete insulin, and they make growth hormone, and they make all these different proteins. But we know that old proteins are broken down. Old proteins are broken down into waste products such as ammonia and urea. NH3 is ammonia. Those are waste products formed from the breakdown of old proteins. What other nutrients do cells need to stay alive? They need nucleotides. Adenine, guanine, cytosine, thymine, and uracil. And what do they do with nucleotides? They join these nucleotides together to form nucleic acids. They make DNA, because you have to do DNA replication before one cell can divide into two cells. They make RNA, messenger RNA, transfer RNA, in order to manufacture proteins at the ribosomes. But we know that old nucleic acids, old DNA and RNA, are broken down. They are broken into a waste product called uric acid. Uric acid is a waste product formed from the breakdown of old nucleic acids. What other nutrients do cells need? They need vitamins and minerals. What is the role of most vitamins and minerals? Most of them act as coenzymes. They assist enzymes in catalyzing the myriad of different types of chemical reactions in the cells of our body. Zinc helps DNA polymerase work. Calcium is needed for ATPase to work. So iron is a constituent of hemoglobin. So these are vitamins and minerals. Niacin is a coenzyme in cellular respiration that transfers hydrogen ions. We also know that water flows into a cell or out of a cell, right? Depending upon how much, what the relative concentrations of water is on the outside of the cell versus the inside. So, we've been learning about cellular respiration. We've been learning about protein synthesis. We've been learning about the role of DNA and RNA. We've been learning about the roles of coenzymes. We have talked about how chemicals can get in or out of a cell. We know that some are small enough where they can just diffuse. We know that when water just simply diffuses in or out of a cell, it's called osmosis. We know that other chemicals, such as sugars, have to be aided across by facilitated diffusion or passive transport. We know that amino acids have to be actively transported using ATP. I wrote at the top, our needs as organisms actually reflect the needs of our cells. It sounds kind of weird. But the reason why we have the organ systems of our body that we do is because of the needs of our cells. The reason why you have lungs is because you need oxygen for cellular respiration and you have to get rid of CO2. That's why you have lungs. You have lungs because cells are all carrying on cellular respiration. They need oxygen. They have to get rid of CO2. If your lungs don't work, you won't be able to get enough oxygen for cellular respiration. You won't be able to exhale, get rid of the CO2, and it will build up and cause what we call respiratory acidosis. The reason why you have a digestive system is because the cells of your body require nutrients, like sugars and amino acids and nucleotides and vitamins and minerals. Those are in your food. That's why you have a digestive system. You digest the food into these nutrients and you absorb them into your body or your bloodstream so that the cells of your body have these nutrients for cellular respiration, for protein synthesis, for nucleic acid synthesis, and coenzymes and so on. The reason why you have a kidney, the reason why you have kidneys, a urinary system, is you have to get rid of these waste products. Ammonia and urea and uric acid are waste products produced by the cells of our body. These waste products are eliminated from our body by your kidneys, by your urinary system. So the point we're trying to make, which sounds weird, is that because our cells have these nutritional needs and they have these waste products that they generate, That explains why we have the organ systems of our body that we do. That's the way modern physiologists explain the role of our organ systems. It is to meet the needs of the cells of our bodies. If we can meet the needs of the cells of our body, our organ systems are doing what they're supposed to. If the organ systems cannot meet the needs, the demands of the cells of our body, the cells will die and we die.